Microbial nutrition. So what does it take to feed a microbe, and why would we even care? Okay, so here the water surrounding this coral reef is dependent on keeping microbial nutrients very low in order to ensure that we keep a balance, that the harmful bacteria do not outcompete the phytoplankton, the other microbes that are necessary, or overwhelm the coral with diseases from the microbes. So microbes are really not that different than humans when we discuss their nutrition requirements. All organisms need certain materials from their surrounding habitat. All living things, need a source of carbon, hydrogen, oxygen, phosphorus, potassium, nitrogen, sulfur, calcium, iron, sodium, chlorine, and magnesium. Now, don't worry, I'm going to give you an acronym to remember that, okay? And it's C. Hopkins Cafe Mighty Good is usually, although this one has a little bit different take than what I'm typically see. This one does have chlorine and not iodine, although those are usually included. So C, carbon, H, hydrogen, O, oxygen, P, phosphorus, K, potassium, N, nitrogen, S, sulfur, Ca, calcium, Fe, iron, Na, sodium, Cl, chlorine, and Mg, magnesium. Those are important materials which are needed for life, all right? Now, the difference is the source of these and the form they come in and the amount needed are going to vary between organisms. Any substance that is required by an organism is an essential nutrient. There are two categories. We have macro, which are needed in large quantities. That's going to be things like carbon, hydrogen, and oxygen. And then micro, which are going to be needed in small quantities. Macro being large, micro meaning small. Those micro elements are also called trace elements. All right. Examples here we're going to have are like zinc or nickel. Those are trace elements, things that we need in a very small amount, but they are very, very important. They are essential for the function of enzymes to work. They're also essential because they are, or the reason they're essential for enzymes to work, I should say, is because they are coenzymes or cofactors. And so they are needed to help form the structure of an enzyme. And so we'll get into that. So before I move on to the next slide, let's discuss what organic means. So stop for a minute, and I want you to go ahead and say it out loud. What does organic mean? I bet I can guess what you said. You said something like healthy or has no pesticides free, no chemicals, okay? Don't get me ranting on that because everything is chemicals. But most people tend to think that. That's what most people think is it means natural or healthy, no preservatives, no chemicals. That's the everyday connotation of the word as we apply it to go into the grocery store, okay? But that's not what scientifically organic means, and so we're going to look at that. So in order to understand what organic means, we're going to talk also about inorganic. So inorganic is going to be an atom or molecule that can contain a combination of atoms other than carbon and hydrogen. So that in and of itself tells you what organic means, which is it's a hydrocarbon. Organic is carbon and hydrogen containing and sometimes oxygen. So inorganic nutrients are going to be found in the Earth's crust, bodies of water, in the atmosphere, and it's going to be things like metals, salts, magnesium sulfate, ferric nitrate, sodium phosphate, gases like oxygen, carbon dioxide, or water, okay? So organic, as I said, is carbon and hydrogen, sometimes oxygen, and they're usually the products of living things. And these are going to range from very small molecules like methane, which is just CH4, to large polymers like carbohydrates, lipids, proteins, nucleic acid. Those are our 4 macromolecules. The source of these nutrients is extremely varied. Some get nutrients only from inorganic sources, and others need a combination of organic and inorganic. And microbes that are very good at being very adaptive at some of the things that they can actually use for nutrients. So while they're very simple structurally, they're very complex metabolically. They're able to use things as a source of nutrition that we could not use. So what are cells composed of? Well, they're about 70% water. After that, the next most prevalent component is going to be protein. 90% of the cell's dry weight is going to be organic compounds. 96% of the dry weight is going to include the elements carbon, hydrogen, oxygen, nitrogen, potassium, and sulfur. And that acronym is pronounced SHNOPS. Okay, if you reverse the N and the O, you could say SHNOPS would help you remember those. Most chemical elements are going to be made available to the cell as a compound, as I've listed in the previous slide, like ferric sulfate, magnesium chloride, not necessarily as a pure element. Only a few type of nutrients are going to be needed, however, for a cell to make over 5,000 different types of compounds. And these are going to be chemical elements that are vitally necessary in order for the cell to grow. So here is another breakdown of that. I'm not going to read all of these individually, but it is really important for you to kind of look at these just to get an idea. So what do microbes eat? Well, that depends on their classification. So we classify a microbe by their nutritional needs, by their source of carbon and the source of energy. So the source of carbon is going to be our category here. Okay, and so you can see we have several different categories. We have autotroph or heterotrophs. Auto means self, and troph refers to feeding, sort of. And so autotroph means basically self-feeder. And so it can take inorganic carbon dioxide for its carbon source. They can actually convert carbon dioxide into an organic compound so they do not depend on other living things for nutrition. A heterotroph must obtain its carbon in an organic form, contrasting that, okay? So next we could look, and we'll look at all of these a little bit more detailed. Next is going to be energy source. Is it going to be photosynthetic or chemosynthetic? All right. Photosynthetic organisms create energy using sunlight. Chemo means from chemicals, so they get their energy from a chemical compound. So heterotrophs, hetero means the same, so a heterotroph gets its carbon from an organic form. An autotroph, auto meaning self, I'm sorry, hetero means different, I said same, it means other. Okay, must get it from something else. Autotroph means self, and that organism uses an inorganic carbon dioxide as its carbon source. It has the ability to convert that carbon dioxide into that organic compound. So again, it's not nutritionally dependent on other living things. So a phototroph would be a microbe that can do photosynthesis. A chemotroph is going to get its energy from a chemical compound. All right, and so we can also break that down into photoautotroph, so light self-feeder, photosynthetic, producing organic molecules using carbon dioxide that can be used by themselves and by heterotrophs. Okay, and then we have our chemoautotroph, so self-chemical feeder. Chemo-organic autotrophs are going to use organic compounds for energy and able to use inorganic compounds as a carbon source. Okay, and I'm throwing a lot at you, but we're going to get into this in more detail. Lithoautotroph, think of lithosphere, meaning the earth substance, relies completely on inorganic minerals and does not require any sunlight or organic nutrients at all. So we have two types of chemoautotrophs here. We've got the chemoorganic autotroph, and that's going to use organic compounds for the energy source and inorganic compounds for carbon. Those lithotrophs are going to, again, like I said, rely solely on minerals and compounds that it can get. It does not need sunlight or organic nutrients. They rely completely on organic minerals. Litho means stone, okay? They also get their electrons from inorganic substrates like hydrogen gas, hydrogen sulfide gas, sulfur, iron, and then they can combine those with carbon dioxide and hydrogen, okay? And so we're going to look at these in a little bit more detail. Most heterotrophs are going to fall under a chemoheterotroph. So they're going to get both carbon and energy from an organic compound. So we have two main categories, okay, which feed on saprobes, which are going to feed on organic detritus, which detritus is waste material from dead organisms. And then we also have parasites that need a living cells or tissues. So here on this slide, we'll look at saprobes. These guys are important environmentally. They're decomposers and recyclers of plant litter, animal matter, etc. Okay. Also, chemoheterotrophs. These are going to get their carbon and energy from those organic compounds, but I don't believe I touched on the fact that these are going to actually take those molecules and process them through either cellular respiration or fermentation. All right, so we got two questions here on this one. I just put them both on one side. Number one, a saprobe derives its energy from A, sunlight, B, conversion of nutrients or other organisms, C, utilizing the tissues or fluids of a living host, or D, metabolizing the organic matter of dead organisms. So pause and come up with your answer. And that will be demetabolizing. The next one is mineral ions used in microbial nutrition include sodium, potassium, calcium, or magnesium. So pause. All right, this one's tricky because it didn't say select all that apply, but it is all of these. Okay, so the next chemoheterotroph was a parasite. Parasites are going to live in or on the body of a host, and they have a symbiotic relationship that harms the host to some degree. All right, so these guys are considered pathogens because they can range from viruses all the way up to a helminth. These are either going to be an ectoparasite or an endoparasite. Ecto means outside, so these guys live on the body. Endo, of course, would mean more inside, and so these live in the organs or tissues, all right? And so we can also classify them as intracellular or obligate, okay? Intracellular, intra means within or between cells, and so those live within a cell. Obligate means they have to. If they're obligated, they must live in a host cell in order to grow. They cannot grow outside of a host cell. An example of some obligate parasites are going to be leprosy, bacillus, syphilis, or even some spirochetes. Okay. Bacteria like Streptococcus pyogenes, which causes strep throat, or Staph aureus, these guys can actually be grown on artificial media. So they would be ecto. So here's a partial list of those essential nutrients. Chemicals that organisms cannot make themselves, right? Those are essential nutrients. So we've got the list of those and where we can obtain some of these. We've got carbon, which can be found from proteins, lipids, nucleic acids, or carbohydrates, which are those 4 macromolecules. Hydrogen is going to be both produced and used by microbes, and it helps maintain pH and forms hydrogen bonds and acts as an energy source in cellular respiration. So it's very important, and it is obtained from water, salt, natural gases. Oxygen is extremely important for enzyme functioning and can also be obtained from inorganic salts like sulfates, phosphates, nitrates, or water, and is about 20% of the atmosphere and can be found in those 4 macromolecules. Nitrogen, atmospheric nitrogen gas is about 79% of the atmosphere's gas, and it is needed, is vitally important for the structure of proteins, DNA, RNA, and ATP, which are nucleic acids. Some bacteria and algae can contain nitrogen from inorganic forms like nitrate, nitrite, or ammonia. Some even do nitrogen fixation, which means they can transform nitrogen into a compound, like nitrogen from the atmosphere, into a compound that is usable by other organisms. Okay, so that's very handy. And this happens in things like the root nodules of certain plants. They live in a symbiotic relationship with plants, a beneficial relationship, where they help the plant roots and then they do nitrogen fixation. All right. So all of this nitrogen must be converted to NH3, which is ammonia, in order to be combined with carbon so that it can make amino acids and other compounds. Phosphate, which is PO43 negative, or phosphoric acid, if it's going to be combined with hydrogen, is needed for nucleic acids, ATP, phospholipids, and some coenzymes like nicotinamide, which is NAD+. So that is extremely important. Sulfur is very important for formation of your B vitamins. Also for amino acids like methionine and cysteine. Those have sulfur or sulfhydryl bonds. All right, so those are very important to help with protein structure, and that's obtained from minerals like rocks and sediment. Metallic or mineral ions are also very important. We only need these in small amounts, but they're very, very important. Potassium, sodium, magnesium, zinc, cobalt, nickel, iodine, things like that. Okay, potassium is really important for protein synthesis and for membrane function. We have things like the sodium potassium pump that is vital to our body. Also for cell transport, calcium helps to stabilize the cell wall and endospores in bacteria. Magnesium is a very important component of chlorophyll and also can stabilize membranes and ribosomes. It's important in our body for things like muscle cell contraction. Iron is important for cytochrome proteins of cell respiration. Zinc is really important regulatory type element that eukaryotes use. So microbes obviously don't have a mouth to chew their food with, and so they have to have a way to take nutrients into the cell and transport waste out of the cell. And so these processes here are basically how they eat, all right? So the driving force of transport is that molecules or atoms are always in motion, and they have a tendency to move from an area of higher density or concentration to an area of lower density or concentration, and they do this without the input of any energy. This is diffusion, okay? So if we look at this picture over here, all right, and we look at the red molecules versus the blue molecules, all right? So, and let's say I have a semi-permeable membrane. The red here is going to be the solute, and the water is going to be the solvent, all right? So let's define that briefly. Solute is always going to be what you are going to have less of. You can say dissolving in, but if it's two liquids, then that kind of doesn't make as much sense. So I like to say the one that you have the less of, okay? And so that's the solute, and the solution is the one you are dissolving in or the one you have more of. So think tea and sugar. So your sugar would be the solute, and your watery tea would be the solution, all right? So here this red substance is the solute, and you can see it is only inside of this sac here that's got a semi-permeable membrane. You can see these little pores here, all right? And so these are going to move in and out of the pore, okay? And so now this is probably meant for this red pore to be a little too large, and so water is usually what's going to be traveling, okay? But let's pretend that these aren't too large to go through. So the red would move or diffuse from a high concentration to a low concentration. So the solute would move out of the little sac here, okay? And so now, as I said, most likely looking at this drawing, these particles are too large to move out. And so what happens is water is what is going to diffuse most often in a cell, because many of the particles can't come out of protein. It's going to be very large, and it's not going to go through those little pores. And so the movement of water through a selectively permeable membrane from a high concentration to a low concentration is going to be osmosis, all right? And so one thing I like to say to help you remember is that water follows salt. So if you think about the fact that you eat salty pork popcorn, what do you want? you want water, okay? And so water follows salt. And when I say salt, I really mean any solute. That can actually be sugar. It can be anything that is considered a solute, all right? And so where do we have more water? All right. So on the outside here, we've got more water on the outside than we do on the inside because we have a higher solute or salt concentration. So what water would want to move in. So the concentration gradient for water is generally going to be opposite of that for the solutes. All right. So don't confuse that. And so we talk about a concentration gradient. All right. So we're talking about moving from a high concentration to a, sorry, I'm trying to do this and I'm kind of over myself here, to a low concentration. So downhill, and that doesn't require any energy. Okay. So think about it this way. If you have a car and you're at the top of a hill, once you get it going, does it take any energy to keep it moving down the hill? No, it doesn't, okay? And so that's going down the concentration gradient. Now, if I'm at the bottom of this hill over here and I want to push a car up, that is going to take a lot of energy. And you'll understand why I'm explaining that in just a moment when we talk about the difference between active and passive transport, okay? And you can also, well, I'm going to get into that just yet. Okay, so when we're looking at how we relate this to cells, all right, we need to define some terms. And these terms are going to be regarding the relationship between cells and their environment, all right? So we can classify the concentration of solution in 3 different ways. We're going to break these words down. So tonic, basically, it means tension. Iso means this. Hypo means below and hyper means above, all right? So let's define some of these, all right? So remember, the solute is what you have less of and the solution is what you have more of, all right? So over here we have an isotonic solution. So we said iso means the same. So the environment is the same in the solute concentration as the cell's internal environment. So we have an equal amount of water. Water will still move in and out of the cell, but it's going to do so at an equal rate, okay? You can kind of think of this like people driving in and out of a town crossing a bridge, okay? And so they leave and go to work in the morning and they come back home every evening. They're the only ones traveling, and so it's going to equal out. It's going to balance out, all right? So there's no net change in the amount of water. It kind of goes at an equal rate there, all right? This top one, we are looking at a cell with a cell wall. So this is going to be much more similar to conditions for things like bacteria. Down here, we're looking at a cell without a cell wall, which is going to be more like eukaryote, which would be like us, or even potentially like our parasites, okay? All right, and so that's isotonic. So isotonic is our happy place. This is where we want to be, okay, for us. Now, not necessarily bacteria. We don't want bacteria to be in this condition. We want to do something to destroy bacteria, right? All right, so in a hypotonic, which is going to be here, hypo means below, so the solute concentration of the external environment is going to be lower than the cell's internal environment, okay? So you can see this in the picture. I've got 2 little water droplets here, or solutes. I'm not sure which they're trying to represent here, but I've got less here and more here, okay? So if my solute, remember that's like my salt, is going to be higher in concentration, water is going to want to go into that to balance that out, okay? So water is going to move into the cell. So net diffusion of water is into the cell, which of course, if you add water, it's going to swell. And so it's going to swell, and in this case, it's going to swell up against the cell wall, which will prevent it from bursting. But if you don't have a cell wall, it actually can cause it to rupture, okay, which is not good or not sustainable for a cell, okay? And so we call this cell lysis that can happen. So hypotonicity, or having a hypotonic environment, is well tolerated by most bacteria because they do usually have those rigid cell walls, okay? So bacteria are a little more okay with that, all right? So in hypertonic conditions over here, this is where the environment has a higher solute concentration than the internal cell environment, which is the cell's cytoplasm. So this is going to force water or draw water out of the cell because of osmotic pressure. And I like to kind of define that as the pull of water. It's the pressure that's wanting water, which way water wants to go. Okay, so this can cause a cell to shrivel or undergo crenation. Now here with the cell membrane here, with the cell wall, that can pull away from the membrane there and cause plasmolysis. Okay, so the plasma membrane there can pull away and rupture, but the cell wall will kind of still protect the rest of the cell area there. Okay, over here, water diffusing out would cause the cell to shrink and become distorted, and we sometimes call that crenation. Okay, microbes do not do well in or they do not perform metabolism well in a hypertonic solution because they are unable to perform metabolic processes. So this is why we've often in the past used salt and sugar solutions to be a food preservatives, and actually they still use these, and I like to use an analogy from my childhood, something you may have done. I grew up in Louisiana, so we had a lot more of them than we do up here, but we had a lot of slugs, which, those little slimy creatures. And what did we do? Well, we would pour salt on slugs. Okay, and what happened? Well, they shriveled up and died. All right, and so that kind of helps you to maybe remember the hypertonic solution. You're adding that salt, water follows salt, it's going to leave the cell because you've poured salt all over the outside of their body. Okay, so just a little morbid illustration there. I don't put a whole lot of these in here, but I think this one is very relevant and very important, and I would definitely read all of these in your textbook, okay? So osmosis and IV fluids. So we're going to look at this situation and see what structure. Well, let's go ahead and read through it first. So the provision of an IV solution is very common in practice. So we often have to give an IV solution. So you have to keep in mind that osmotic movement of water occurs while the body's trying to, of course, balance out or create a balance between different solute concentrations that exist on either side of that semi-permeable membrane. Okay, so we can have some different types of IV solutions that are used in medicine. Isotonic solutions have tonicity that is the same, of course, iso meaning the same, as the body's plasma. So when these are administered, there's not going to be a ton of movement, if any, between the tissues and the blood vessels, all right? Hypertonic solutions are going to have, of course, a tonicity that is higher than that in the body. So administering a hypertonic solution will cause water to shift from those extravascular outside the vessel spaces into the bloodstream, which will increase the volume of those vessels. All right. And so this is how the body attempts to dilute the higher concentration of electrolytes in the IV fluid. And again, that can be salt, it can be sugar, potassium, different things. Okay. Hypotonic solutions have a tonicity that is actually lower than plasma, causing water to shift from those blood vessels for the intravascular, actually between blood vessels, to extravascular space, which means outside of the blood vessels there, and eventually into the cells and tissues. And so in this case, the body's going to move water from that intravascular space to the cells in order to dilute the electrolytes in the cells. So what structure here then is the semi-permeable membrane? Well, for IV fluids with different tonicities, the structure we're talking about is, of course, those blood vessels wall. All right, and those are going to exclude larger solutes but not water. So it's always going to be the measurement of what goes through is going to be determined by whatever that semi-permeable membrane is. Transport processes in cells. This is really pretty important to understand. I talked about this a little bit about whether or not we needed energy earlier when we were talking about going uphill or downhill. That's a very important distinction. We want to be able to classify things as either active transport or passive transport. Okay, and then we have different types in those categories. Okay, so differentiating between that active and passive transport, we have to look at one, whether or not energy is needed to move molecules in or out of cells. That's the most important way. Also, then, are you going from a high to low concentration or low to high concentration? Are you going downhill without any energy, or you pushing uphill, and so you need that extra energy, okay? So the first one here we have is going to be our passive classifications here. Osmosis would also fall under here. I think this is broken down into 2 slides. I don't know if it's just not included, but basically osmosis is just diffusion of water. It's just specific to water, okay? So small substances like non-polar molecules, lipids, or lipid solid molecules can pass through the membrane, because remember, the membrane is a phospholipid bilayer. Then that is going to regulate what is able to go in and out. We need to be able to move polar molecules and ions across the plasma membrane, and so then we have developed facilitated fusion and other active processes to help them move across, okay? So diffusion, simple diffusion, is a type of passive, so you are going from high to low concentration. Let's get a pen here. So we are going from high to low concentration. You are going down that concentration gradient. So no energy is required. And this is just going to occur because of that concentration gradient. Okay, so atoms are going to typically go from a higher concentration to a lower concentration on their own. It's what we would call a spontaneous process in chemistry. It's going to happen without the input of any energy. It's just going to occur, okay? Now, facilitated diffusion you want to watch out for, because this is the one most frequently misunderstood, because you're thinking because it's using this carrier molecule here that it is active, but it is not. So you got to pay attention to high concentration to low concentration. So facilitated diffusion just means you need a little extra help getting in, okay? And so a molecule might bind to a receptor here in the membrane, okay? And the molecule will go across. And so transport is limited by binding sites of these transport proteins. So you have to have this transport protein here, but it's still going to occur without the input of any energy, okay? So En, there is no energy needed. And I could actually, instead of energy, I should say ATP, because that is our energy currency, right? No ATP, no energy is needed, okay? So simple diffusion goes on its own from high to low without the input of any energy. Osmosis is diffusion from high to low without the input of any energy of water. And facilitated diffusion, you do have a carrier protein. I probably shouldn't say carrier. You do have a transport protein would be the correct terminology there. A transport protein embedded in the membrane that these will use to go through. So it's kind of like an automatic doorway, okay? This guy, no doorway is needed. It can just go in and out without a door. This one is one of those automated doorways. You don't have to push the door open. It opens up for you automatically, okay? And so still you're going from high to low, no energy needed. Now, active processes, you're going uphill. So you're going to have to expend that energy to push that car uphill, okay? And so here is the carrier molecules. That's why I corrected that. These are where you're going to have carrier molecules. This is often going to be things that are going to be what we call pumps, like the sodium potassium pump. All right, and so ATP is going to drive the pump to move. We call that proton motive force, and that is because it's driven by the movement of protons, but that's kind of getting way ahead, kind of technical, all right? And so what you need to know is that this is going from low to high concentration, okay? And so we are now going uphill. Okay, so we're going from low concentration to high concentration, all right? These are going from high concentration to low concentration. So I'm working here because I'm pushing something uphill. So I added this slide mostly because of the pictures, but I went ahead and drew it for you, which I know would have. But anyway, so transport of nutrients against the concentration gradient. That means going uphill. You can see that here. We are going uphill, or it's going to require energy. If it's going in the same direction as the natural gradient but needs to be sped up, you could also do this, but we mostly are going to look at it this way. We're going from low to high, okay? So we have specific proteins, those pumps, and then also some permeases. that are involved in this. And of course, I've already said we have the expenditure of energy. Some examples of things that are transported this way are going to be monosaccharides, so some simple sugars, amino acids, organic acids, phosphates, middle ions, all right, those types of things, because they're charged. Some of them are charged anyway. So features of active transport that are really important to remember is you're going against the concentration gradient. So you're going from a low to high, and you can see that really well in this picture here. You're going to have a specific membrane protein often, and you're going to expend some energy, okay? All right, that's it. All right, so here's some more examples of basically cell eating and drinking. All right, so movement of substances in ways that aren't necessarily going across the membrane. This is a little bit different here. We have large molecules that cannot physically pass through the membrane, and then so we have to carry them inside via endocytosis. Endo meaning inside. We also have exocytosis, which is the reverse opposite, which means things exiting the cell. Okay, so endo inside, large molecules that cannot pass physically through the membrane can be carried in via endocytosis. And so here we are looking at two different types. In endocytosis, the cell is going to enclose a substance in the membrane, form a vacuole, and engulf the substance with the membrane. All right, and the main difference of these two here is going to be whether or not it is solid or liquid. All right, so we have phagocytosis over here. Phago basically means eating, and so this is cell eating. And so these are solid particles that are being taken into the cell, engulfed, and then digested. All right. Things that do these are going to be microbes like amoebas. All right. So this is one way that they are eating. And then our body uses them like white blood cells is actually part of our defense mechanism. Okay. So they're ingesting whole cells or large solid matter. And then we have pinocytosis. And the way I try to help students remember this one is Pinot. And I'm not really much of a wine drinker, but you see commercials about Pinot Noir, or Pinot Grigio, and it's a type of wine. And so maybe that will help you remember that it is drinking. Pinocytosis is cell drinking. Now, there may be small solid substances that are or molecules that are dissolved in the solution, but it's in a solution, so it's liquid. All right. And so things that are small like that will enter through pinocytosis.